Namaste to all. Welcome to the next session on molecular biology and our DNA technology. In this session, we are going to see about DNA replication in prokaryotes. So, as we have seen in our previous session, how the origin of replication fork is produced and what are the different types of replication which takes place in prokaryotes as theta model and rolling circle model. So in this, we are going to see in detail how the DNA replication is carried out. So as I said, it is a continuous process and there is no division like initiation, elongation and termination. It is a continuous process which is taking place. Along with that, the transcription and translation also continues. But for understanding, we are dividing the phases into three as initiation, elongation and termination. So if you see the initiation phase, so this is our DNA strand which is present. And in this prokaryotic DNA, that is the circular DNA, there are four sites which are present here, which is called as DNA A. Each site has 9 bases here. And upstream of this, they have 3 sites which have 13 bases and they are 80 rich sequences. So this 9 that is the 9 base pairs containing 4 sites are called as DNA A box. So this A box is also acting as origin of replication. So in prokaryotes, we say this as ORI-C, and in plasmid or vector, we call them as ORI-V. So they have 4 sites having 9 bases per site, and they are called as DNA A box, initiation recognition sequence, or origin of replication, that is ORI-C, as I said, in prokaryotes, and ORI-V in vectors. Upstream, that is 13 base pair long sequence and having 3 sequences, that is 80 rich region, site where the replication actually originates. So to begin this process, what is happening is, so we have DNA A. This DNA A molecule or proteins or factors, they will come and bind at this 4 site. Once they come and bind at this 4 site, because of the complementarity nature and the attraction, they fold and become circularized. Once they become circularized, all the 4 sequences are present in all the 4 sequences are actually seen, and the 13 bases containing 3 sites will be exposed. So due to this, what happens is it creates tension, and due to the circular DNA and causing a negative supercoiling, what happens is to release this tension in the upper upstream of this particular location, that DNA melting occurs in the sequences. So due to the DNA melting, what happens is the hydrogen bonds between the adenine and thymine, cytosine and guanine, they starts to divide. But since in the upstream 13 sequences, they have 80 rich sequences, and between adenine and thymine, they have double bonds of hydrogen. So the breaking of this is very easy. Due to this reason only, in the upstream, the ATP, the adenine and thymine, they break and the DNA melting takes place. So with the help of ATP hydrolysis, that is becoming as ATP to ADP and PI, in this particular region, what happens is the DNA B and DNA C, they join together. So DNA B and DNA C, when they join and form a complex, this is called as helicase. And this helicase allows the DNA melting at the upstream position. So the DNA B, they cannot melt the DNA on its own. DNA B is 6 subunits, and DNA C is also having 6 subunits. They form the complex, that is DNA B and DNA C complex, which is called as helicase and which helps in the melting of DNA now. So due to the melting of DNA, you can see here the DNA B and C complex loaded in one of the strand. It is not in both the strand. It is loaded in one of the strand of the DNA. and they start to melt the hydrogen bonds which are present. Once the hydrogen bond is removed, now what happens is they tend to form the bubble, and the replication fork is formed here. So in the 5 prime to 3 prime and 3 prime to 5 prime, so it is about to take, I'm going to take it only one side where the replication fork has started producing, and it is an anti-clockwise direction, and the direction of the movement is basically in 5 prime to 3 prime direction. That is, from this part to this part, this is acting as our leading strand, and the other strand which is there, that is going to be our lagging strand. So now what happens is, once the melting has taken place and origin of replication fork has started and bubble has formed, now what happens is the single stranded stabilizing protein or binding protein, they will come and bind, which does not allow the recoiling of the particular strand. And after the binding of these strands, what they do is now they bring the primase. Basically, this primase we call here as DNA G. So what happens is, once the primase comes, the one of the part of the single stranded binding protein gets removed. And now what happened is we have our DNAG, which is primase, and they bring few RNTPs at this particular location. Once they bring nearly like 5 to 10 nucleotide sequence when they are being formed in the 3 prime end, now what happens is the primer is being synthesized, which is this primase is actually DNA dependent RNA primase. So they are RNA polymerase. So they are bringing the RNA here, and after bringing the RNA here, now what happens is they can initiate the process of elongation. So in the process of elongation, we can see here already the primer is ready. And now we have to have, this is our leading strand. The other strand is our lagging strand. So to make the lagging strand, what happens is, to undergo the elongation, the other strand, the bases, actually the single stranded binding protein gets removed, and they form the replication loop here. And in this strand also, primase binds and they add the primer at different location. Once the primer is added at different location, now this is our leading strand and this is our lagging strand, which is having the replication loop here. So once the polymerase is binded, they can move to and fro, and they can produce the particular strand. So if you see in this process, now what happens is, since we are having this already ready, so the single stranded binding protein starts to remove and they move towards the anti-clockwise direction. So this is our RNA polymerase. So once the RNA polymerase come, they bind in both the loops, that is in the 3 prime to 5 prime direction, and the other loop, because of the RNA, that is the replication loop formation, that has also formed as 5 prime to 3 prime direction movement. So now what is happening is, since the loop is present, so it has to adjust itself. So once it moves, again stops, again pulls, again makes another one. So that is why the lagging strand, they will have the nick or breaks in between, and that is why that is called as Okazaki fragment. So now the DNA polymerase, which is the DNA polymerase 3, which is present here, now it starts to move in the anti-clockwise direction or in the 5 prime to 3 prime direction. So once the RNA polymerase moves, they also have the exonuclease activity. So exonuclease activity means if any wrong base has been added, they can remove, because consider this is your polymerases, they have the exonuclease activity. So if any wrong base is added, they can remove that base and they can do the proofreading also. So this is how the strands have been synthesized. So it is moving in this direction and it is being synthesized. So now since the strand has been synthesized, now what happens is, to remove the primer, we have another enzyme here, and this enzyme is called as RNase H. So this RNase hedge removes the RNA primer, and now it brings again the RNA polymerases, and the RNA polymerases again synthesizes the DNA strand. And since you can see here, there is a small gap or a nick which is present here. So to fill up this nick, we have to use another enzyme that is called as DNA ligase. So DNA ligase, what it does is it will bind the it will bind the nick or fill up the nick, and it will make as a single strand. So the RNA is degraded and the nick is formed here. So the DNA ligase, what it does is the DNA ligase joins and it becomes as a normal strand now. So to see this, the bottom one is our parental strand and the red color one is our daughter strand. So let's see the DNA ligase activity, how the DNA ligase works. So if you see, this is the nick which I was talking in the previous slide. So here, if you see the nick, they have the 3' OH and 5' phosphate end. So the 5' phosphate end, if you see, the phosphate molecule is present, and the DNA ligase, when it comes, they bring the ATP molecule along with it. So what it does is, once the DNA ligase binds at this particular location, mainly at the 5' end of this nick, what happens is the O group which is present, they have more affinity towards the alpha phosphate of the ATP. So what happens is, due to the nucleophilic attack at the alpha phosphate, they remove the PPI, and the AMP molecule is joined at the 5' end now. Once it is being attached at the 5' end, you can see that it has now become as alpha, and now this has become as beta. So due to this, the OH group at the 3' end has more affinity towards the alpha phosphate. So at this particular point, because of the nucleophilic attack, they break. Okay, so they attaches with this and they break the AMP or AMP molecule, and that gets removed by the ligases. So like this, the nick is actually joined here and formed as a single strand. So the phosphodiester bonding has taken place and the nick is removed. So let's go to the termination now. So in the termination process, so as we have seen that the theta model has started developing, the strands are forming in the clockwise and anti-clockwise direction. So now what is happening is there are two sites which are present here, which are called as ter A and ter C, that is for termination. This sites, this is our replication bubble, and the ter site, if you see, that is 24 base pair which is present here. It is present at multiple locations and helps in the termination of replication. So what it does here is, so during this process, I am taking only one strand which is present here. So which is having our ter site, that is ter A and ter C. So now there are two factors which can come and bind, that is called as TUS. This TUS is actually termination utilization sequence. So this TUS actually has a protruding domain which is present here. So this protruding domain is like a measurement, or you can say the signaling aspect for the replication termination. Once this TUS factor comes and binds as ter A and ter C, the direction of the protruding region actually plays a vital role here. They have the, if it is actually present like this is called as permissive ends, and if it is not joining, then it is a non-permissive ends. So let's see how it is working. So in this process, if the fork touches the TUS, that is the TUS ter permissive ends, then what happens is it removes the complex and the fork actually moves. So you can see this is this particular end, that is the plain end, what you see here, this is our permissive end. And if the fork, that is replication fork, is coming and it touches here, the TUS is actually removed and it can move forward. That replication fork can move forward. The non-permissive end is like a protruding end. So what happens here is, when it touches, the fork encounters the tuss or tur non-permissive end. It does not allow the DNA B and C complex, that is helicase, to move forward, and the replication is halted or stopped. So the flips one or two bases, and usually the flips multiplies cytosine bases, that is C. So you can see in this particular diagram how this is working. So consider this is our, in one strand we are having our helicase which is present, that is DNA B and C. This is our ter C. We have already seen in our previous slide, it is having the non-permissive end which is at this particular moment. So what happens is when it comes near to this region, what it does is it flips, okay, the cytosine which is there nearby, it flips the cytosine towards top region. So since it is flipping the cytosine to the top, that is upper part, what happens is the DNA helicase cannot move forward and creates one-way direction for the helicase movement, leading to the termination. So I hope that in this video you have got an idea about how the DNA replication takes place in prokaryotes, and what are the different methods, that is initiation, elongation, and termination, how it takes place in prokaryotic DNA replication. See you in the next session with eukaryotic DNA replication. Thank you and have a good day.